Title of my paper is A Neuropilin-like Protein, ESDN, Regulates Insulin Signaling and Sensitivity. Just to give you guys a little bit of background on insulin, it's a peptide hormone that's produced in the pancreas by the beta cells. It's involved in the homeostatic regulation of blood glucose by inducing the uptake of glucose from the blood into the cells. It does this in a number of ways in different parts of the body, namely by inducing the absorption of glucose by the intestines, regulating the uptake of glucose into the skeletal muscle cells, inhibiting the production of glucose in the kidneys, and synthesizing glycogen. It's also found to be involved in the differentiation and growth of the cells, and one of the major problems that's associated with insulin signaling in humans is insulin resistance. So a number of the different regulatory proteins involved in insulin signaling have been examined as potential targets for therapeutic drugs. To kind of generalize what's going on in insulin signaling, to begin, insulin enters the bloodstream and it binds to the insulin receptors found on the surface of adipocytes, hepatocytes, and skeletal muscle cells. Binds particularly to the alpha subunit of the insulin receptor, which is the extracellular domain. This then activates the tyrosine kinase domains on each beta subunit of the insulin receptor, which then leads to the phosphorylation activation of two different pathways, the MAPK and PI3K pathways. The MAPK pathway, when activated, leads to the growth of cells as well as gene expression. And the activation of the PI3K pathway is involved in the synthesis of different molecules like proteins, lipids, glycogen, and is also involved in cell proliferation. ESDN, which is the main focus of my paper, it stands for endothelial and smooth muscle cell derived neuropilin-like molecule. Kind of a mouthful, so I guess that's why they call it ESDN. It's a transmembrane protein that's encoded by the ESDN gene and is expressed primarily in the liver, skeletal muscle, and adipose tissue, which is also where the receptors for insulin are found. It's involved in the vascular remodeling and angiogenesis via VEGF and PDGF protein signaling in vascular cells. VEGF stands for vascular endothelial growth factor, and PDGF is platelet-derived growth factor. So what does ESDN have to do with insulin signaling? It's found to have inhibitory effects on both VEGF and PDGF protein signaling by interfering with receptors for both of these proteins. And like VEGF and PDGF receptors, insulin effects are mediated by a receptor tyrosine kinase, which is located on that beta domain. So if VEGF and PDGF signaling is modulated by ESDN, then it's also possible that ESDN is modulating insulin signaling. The main purpose of the experiment that they conducted was to determine the role of ESDN in insulin signaling and to determine how insulin signaling adapter proteins interact with ESDN. So kind of getting into the mechanism involved with how ESDN interacts with the proteins in the insulin signaling pathways. And they also wanted to determine how the loss of ESDN affects the metabolic effects of insulin. The overall experimental design, they worked in cultured vascular smooth muscle cells, and they targeted the inactivation of a number of genes, namely the ESDN gene. And they also used the administration of insulin to these cells, either with or without ESDN. The other tests that they performed were the glucose and insulin intolerance and mouse body composition tests in order to determine ESDN's role in regulating the metabolic effects of insulin. And finally, they performed a number of co-immunoprecipitation studies in order to identify exactly what's going on between insulin and ESDN and the insulin receptor and adapter proteins. The first figure shows you the results of the transwell migration assay, which was used to determine exactly how insulin signaling is being impacted by the loss of ESDN, which is what you can see on the right side of figure B. And what you see is in the cells that lack ESDN, when an insulin is being administered, you get a much more enhanced migration than the cells that do express ESDN, which is the wild type. And figure C is just showing you the representative quantification of the data that you see in figure B. The second figure here shows you the effects of ESDN deficiency on the phosphorylation of the important insulin signaling molecules like the insulin receptor, the MAPK and AKT enzymes, which are involved in the MAPK and in the PI3K pathways. And what you see is in E and F, after 10 to 20 minutes of administering insulin, the amounts of these phosphorylated proteins, MAPK and AKT, are significantly higher in the ESDN deficient cells than the cells that do express ESDN. And in G, you see after 20 minutes of administering insulin, the insulin receptors that are phosphorylated are significantly higher in the ESDN deficient cells. From these first two slides, we conclude that ESDN deficiency is enhancing the signal transduction of insulin in cultured VSMCs. ESDN deficiency is also significantly enhancing the phosphorylation and activation of important molecules involved in insulin signaling, molecules like IR, MAPK, and PI3K. This shows you that it's affecting both metabolic and mitotic effects of insulin in cells. This next figure is a insulin tolerance test in wild type mice and ESDN deficient mice. And what they measured was the blood glucose levels in each of these mice over the course of 120 minutes. And what you see is that after roughly 30 to about 80 minutes of administering insulin, the ESDN deficient mice have a much lower blood glucose level than wild type mice. Next, they knock down two of the adapter proteins that are associated with insulin signaling. So the proteins in this case are APS and GRB10. And you can see in figure F that after inducing with insulin, when they knock down APS, there's a significant decrease in the phosphorylation of the insulin receptor in both the wild type and the ESDN deficient mice. And in H, when they knock down GRB10, there was a significant increase in the phosphorylation of insulin receptors in the wild type mice, but decreases in the ESDN deficient mice. So from these figures, we conclude that ESDN deficiency is enhancing the sensitivity to insulin, and that the APS insulin receptor interaction is mediating the ESDN effect on insulin signaling. Also, the GRB10 insulin receptor interaction is inhibiting the ESDN effect on insulin signaling. From the results of the data that they collected, they proposed a mechanism to show you how ESDN is affecting the signaling pathways involved in insulin signaling. So you can see that insulin is binding to the insulin receptor, and ESDN is inside the cell inhibiting APS, which leads to the ubiquitation and internalization signal pathway. ESDN is also inducing GRB10, which is inhibiting this ubiquitation and internalization signaling pathway. So in summary, just to kind of give you guys an idea of how this would apply in the clinical sense, patients that have type 2 diabetes and insulin resistance, ESDN is most likely expressed in higher concentrations because it's preventing insulin from reaching the insulin receptor, and it's not allowing insulin to enter cells from the bloodstream. So it's possible that ESDN, or the proteins that are associated with ESDN, could be a potential therapeutic drug target for patients that suffer from these symptoms.